Thank you very much for this on behalf of the Institute. This grant has actually made a significant difference to the way that we actually do our science. As Tom said, the money's been used to actually buy high-tech equipment, and we've actually gone out and bought some equipment which is actually crucial in our goal to try and understand the genetics behind some of the cancers that we actually see in Tasmania and the rest of Australia. So we've called this the Tasmanian Inherited Cancer Centre, and the word inherited is crucial here because what we actually study is cancers that actually run in families. And this is an example of some of the families that we look at. And this is a particularly Tasmanian approach to studying cancers. It's not Tasmanian for any reason at all, except that we actually have a fairly unique social environment here in the state, where there was a rush of immigration in the early 1800s that essentially petered off by the mid to late 1800s, and about 60 to 65% of the current day Tasmanian population is related to those early settlers back in the early 1800s. What that means is that we have some enormous pedigrees from these original immigrants that came in the early 1800s, have actually produced children, have produced children, have produced children, and we've actually got up to 10 generations of Tasmanians that now live in this state. Many of them are still here. There's been very little exodus and very little ingress as well. So a large percent of Tasmania's population at the moment is comprised amongst these very large pedigrees. What we can actually do is chase down these pedigrees, and we've got some very talented genealogists, and they're around here somewhere, who actually go around and put together these pedigrees so that we can actually find large families that actually have enriched for a particular disease.
Okay, all right. Right on. So we now, I think, officially declare Tasmanian Inherited Cancer Centre open.
We had to wait for the rep to come down. And, yes. We have to do it all according to specifications.
Can you see that the robot's got one of the displays in the building?
I know!
Cool to -10 through to 95 degrees Celsius. Shake at the same time.
This little beast is here. This is a laser dissection microscope. And so what you can do with this microscope is actually look at slides and cut out the smallest amount of section that wouldn't be physically possible with the human eye, and then collect that into a tube that you can actually extract DNA from later, or whatever processes you want to do after that. So Liz, who Simon was talking about this morning, who's got the hematological malignancy, she actually cuts out little tiny sections of chromosome. And what I'm showing you here is actually some of Joe Dickinson's work with the prostate cancer cells. So this is a normal prostate tissue, and you can see here that the lumen look quite nice, well formed, and everything looks extremely pretty until you look at what a prostate cancer cell line actually looks like. The cells grow so fast it destroys that lumen and the structure, and so these cells sort of just invade and take over. What we couldn't have done before was actually separate this tissue from the healthy tissue, which you can see in the corner of this slide. So you'd be extracting DNA from cells that are actually cancerous and healthy all in one sample. So this way, what you can do is you can actually choose a section that you want to actually cut out. So in this case, you would cut out this section here, and you would extract your DNA from that and do all your sequencing on that. And you can actually have normal tissue from the exact same patient. So you're actually comparing against self. So what you can do is cut out a section on here, any shape that you like. And what we'll do is then turn on the laser, and the laser will actually follow the line that you have actually done an outline of. Then hit the slide really, really quickly and catapult that section you've cut out into the cap of this little Eppendorf tube here. So one that I've done previously you can actually check that the cells would have actually gone up into that cap by moving that cap over on top of the microscope. And funnily enough, there's a little R